In this lesson on membrane structure, we're going to focus on membrane fluidity, and you'll be able to outline the importance of fluidity to the function of the cell membrane, as well as explain the effect of each of several parts of the cell membrane to its fluidity, including cholesterol and fatty acids. First, the cell membrane needs to be fluid to support movement of the cell membrane, as well as movement of the cell overall. Permeability, so the ability to be selectively permeable, let some things in and some things out, that's going to depend on membrane fluidity, and a lot of that has to do with protein function. There are proteins embedded in the membrane that help to regulate what goes in and out. There are also other kinds of proteins in the membrane, like enzymes, and their function depends on membrane fluidity as well. We will learn later about kinds of transport called exocytosis and endocytosis, or you might have encountered them already, and they very clearly depend on the membrane moving in order to engulf or release particles. So how does the membrane stay fluid across a variety of different environmental conditions, for example, temperature? Well, in animal cells, one factor is cholesterol. Cholesterol is only in animal cell membranes, and cholesterol acts as a fluidity buffer for animal cell membranes. A buffer is something usually when we're talking about acids and bases that keeps the pH within a narrow range, and cholesterol acts like that for fluidity. At high temperatures, when there's a lot of molecular motion, and so the membrane would naturally be getting more fluid, cholesterol decreases fluidity, so keeps it closer to its fluidity at more moderate temperatures. And it does that by restricting the movement of phospholipids. I'm going to write PL for phospholipids. It restricts the movement of the phospholipids laterally in the membrane as it intersperses with them. Remember that if this is our phospholipid bilayer and we've got integral proteins and peripheral proteins, then cholesterol is going to be interspersed in between, here's cholesterol, interspersed in between the phospholipids, and that can restrict the lateral movement. In contrast, at low temperatures, when the membrane would usually be getting less fluid as there's less agitation due to thermal movement of particles, in those situations, cholesterol increases the fluidity, again back towards its fluidity at more moderate temperatures. And the way that it does that is that it prevents the close packing of the phospholipids. So if you take a look at the phospholipids in this diagram here, they would like to get close together to each other, to have the hydrophobic tails next to each other. But if cholesterol gets in the way of that, it prevents them from getting close and effectively prevents them from freezing, if you like. So by restricting phospholipid movement at high temperatures and preventing close packing of phospholipids at lower temperatures, it maintains a more constant fluidity of the cell membrane in animal cells. Let's look at another mechanism that affects fluidity. Double bonds in the tails of unsaturated fatty acids also interfere with close packing of phospholipids, like we saw with cholesterol above, and so they also increase fluidity, like cholesterol did at low temperatures. So a fatty acid has a carboxyl group at one end that I'm not going to draw, and then it has a hydrocarbon tail, meaning lots of carbons joined together with hydrogens around it. We'll get into the chemical structure of fatty acids more in a later unit, but for now, this is the hydrocarbon tail part of it. Carbons with hydrogens. Now this is an example of a saturated hydrocarbon, or a saturated fatty acid here, meaning that there are only single bonds between the carbons and it has as many hydrogens as possible. That's the saturated part, saturated with hydrogen. And saturated hydrocarbons are able to pack closely together because of their shape. In fact, most animal fats are made of saturated triglycerides, which have saturated tails like this. And that's why they are solid at room temperature, like bacon fat or lard. In contrast, if we put a double bond in there, something like this, that's going to create a kink that then prevents close packing of this hydrocarbon tail, this fatty acid, with this one. And so this is an unsaturated hydrocarbon. Again, if I drew the carboxyl group at the top, we could turn it into a carboxylic acid. So this is an unsaturated hydrocarbon, and it doesn't pack closely next to its neighbors. And so that's going to increase fluidity. Think of it as moving away from freezing, if you like. And most vegetable oils contain unsaturated fats. Like this prevents close packing. They are liquid at room temperature. The same is true in phospholipids. If phospholipids have a double bond in the fatty acid tail that creates a kink like this, because it's unsaturated, as drawn here, they're not going to be able to pack very closely next to their neighbor, and so that's going to increase fluidity. In contrast, saturated fatty acid tails are going to allow the phospholipids to pack closer together. And that's going to decrease fluidity compared to the above. Last thing, the longer the neighboring fatty acid tails are, the more opportunity there is for them to interact with each other. So if I increase this length here, now there are more atoms that can attract each other by intermolecular forces. And therefore, this is going to have lower fluidity than in the shorter case above. So the longer the neighboring fatty acid tails are, the greater the intermolecular forces, I'm going to write that as IMFs, are between them, and therefore the lower the fluidity. So longer fatty acids result in lower fluidity because there are more attractive forces in between the fatty acid tails holding them together. If you study chemistry, then the specific forces are London dispersion forces that are happening between the hydrocarbon parts of the fatty acid tails. Okay, so that's an introduction to membrane fluidity.